Our next speaker is Hongxia Fu, who is an assistant professor in the Division of Hematology Oncology, Department of Medicine at the University of Washington. And she's a member of the Bloodworks Northwest Research Institute, who a whole group of them have joined us today, as Dr. Conway has mentioned earlier. Hongxia obtained her PhD at the National Institute of Singapore, where she studied mechanobiology using single molecule biophysics tools and theoretical models, and that's why you asked that really good question earlier. She conducted postdoctoral training in biophysics at the National Institute of Singapore and in cellular and molecular medicine at Harvard Medical School. Her research focuses on understanding the mechanisms of hemostasis and thrombosis, seeking new therapeutic strategies, and thanks very much for making your way up in the rain. Thanks.
Thanks for the invitation. And so I will talk about the FVIII VWF interactions from the single molecule studies. So VWF and FVIII are very important to the thrombus formation, and VWF can bind to platelet, and also VWF is a carrier protein for FVIII. So VWF is a multimeric polymer in blood, and I just want to mention the D' D3 domain on VWF actually can bind FVIII and A1 to the platelet GPMB alpha. So we all know the flow-induced hydrodynamic force actually can regulate the thrombus formation, especially for the diseased case like stenosis or the bleeding. So a lot of studies have already been done to understand VWF FVIII interactions, including Dr. Lawless' work. Most of the work actually is used bulk preparation of VWF. So the questions we want to ask is how can FVIII interact with individual VWF monomers, and also the effect of the flow, and also the interplay between the GPMB alpha and FVIII. So to answer these questions, we actually use a single molecule fluorescence imaging and the microfluidic system. So this is our actual system. We use the recombinant VWF. So we label the Alexa fluorophos 488 so we can visualize it. And also we label with the biotin so we can attach VWF on the flow channel surface, which actually coated with streptavidin, and we can add the flow from different directions and also adjust the flow rate. So using this system, you can see the VWF actually, the green is VWF multimer attached on the surface, random coil structure, and then we add the flow. So the flow we use the shear flow as an example here. So we adjust the flow can elongate VWF, and when we add in another fluorescence labeled FVIII, which is Alexa 647 labeled, and you see the FVIII start to bend on this VWF monomer, and after a certain time, usually about like 15 to 20 minutes, you will see the saturation of FVIII bending, and then after that, we add buffer to wash out the free FVIII and see the dissociation. So we found the FVIII band to VWF monomer they have the KD around 0.3 nanomolar. And when you increase the shear stress, then you can see the slightly increased KD. And we found the FVIII can evenly bind to individual VWF monomers. That is on the left page figure here, and also this binding is independent on the shear tension along the VWF. So we also found even though we wash out the free FVIII from the solution, then you still see the FVIII band on the VWF, which is here. So only for example, if we use the thrombin or high concentration of the calcium to wash out, to add into the system, then you can see the FVIII most affected left VWF. So we can see the electrostatic interactions actually played an important role in this bending. So what this tells us, so we know in the bloodstream, the free factor VIII can be degraded very quickly. So that means if you're adding a bunch of the factor VIII into the system, then actually a lot of factor VIII will be degraded. But if you add in the optimized ratio between the factor VIII and VWF, which only shows have the strong interactions between them, so that means it's hard for them to disassociate with each other. So we found for this strong interaction between VWF factor VIII, it can stay in our experiment channel for days. Basically, it's still there. So the next question we want to ask is how the GPMB alpha and factor VIII interact, interplay with each other. So from our previous study, we found GPMB alpha, to initiate the GPMB alpha band with VWF, you basically need to have a critical tension on the VWF, which is around 21 piconewton. So what we did here is we pre-bound the factor VIII on VWF, and then after that we add in GPMB alpha. Then we add flow, and then we see the binding of GPMB alpha on VWF. We found we need higher tension to initiate GPMB alpha binding on pre-bound factor VIII VWF. And also, we have lower number of the GPMB alpha binding on the VWF. So that means pre-bound factor VIII actually can kind of decrease the GPMB alpha binding to VWF. But the GPMB alpha has no effect on the factor VIII binding on VWF. So in summary, in our study, we use a single molecule experiment method to find factor VIII band on evenly along the VWF multimer with KD around 0.3 nanomolar in the static condition, and also tension is does not play plays a role in the VWF factor VIII binding, and also electrostatic interactions play an important role for the VWF and factor VIII strong binding interactions. And the pre-bound factor VIII actually can increase the force to initiate the VWF factor VIII binding. So this is our model. So we suspect maybe because D prime D3 one are so close, so factor VIII bind to D prime D3 may inhibit or shield A1 domain to bind with GPMB alpha. So at the end, I want to thank our lab members and also collaborators and the funding agencies. Thank you.
I'm curious how you're chemically modifying the von Willebrand factor and the factor VIII with those fluorescent probes, if the chemical modifications are homogeneous and if they are influencing in any way the interactions.
Okay, so that is a very good question. So we basically use the kit from the company, and so labeling is just a random label on the amine domain of the VWF. So after that, we measure the degree of labeling. So the labeling ratio is about 0.7 fluorescence per VWF monomer. So based on our so this kind of low ratio of the labeling, so I don't think they have much of the effect on the VWF function. And we did the ELISA with the control for the GPMB alpha binding. We didn't see much of the difference. Yeah.
In your fluorescence images, BWF starts off as a kind of a spot, and then under flow you show this streak of BWF. Is that like BWF strings of several BWF molecules strung together, or is that just a single molecule that is producing that image?
Yeah, so So that is a single VWF monomer. Is the reason because we actually calibrate the single fluorescence and also the degree of labeling and measure the fluorescence intensity along the molecule. Then we know actually like a single molecule fluorescence, basically is around 0.7 each fluorescence dye along the distance along the monomer. Yeah, so we think it's a single molecule. Good question.
Thank you.
Yeah. Yeah, so we also use this one to study self-associations, and you can see basically you can count the number of the associate, not the color of the VWF on it.